little. We don't want to see me.
That's right. We have to do that on our own.
Yeah, that's fine. Can you see my map, my pointer?
Yep.
Yeah,
right. Yes.
great.
Perfect.
Just make sure I've turned my phone off so my mother doesn't ring. Trouble is, if you turn your phone off, she rings the house phone.
Right. And yes, no, you can't do much about that.
No.
Have you got your students back in the lab?
No, he's safely at home, aren't you, mate? We've got our ethics is suspended at the moment because all face-to-face ethics was stopped. So there's an ethics meeting, I think, on Friday. They've said I might need to be on standby to speak to the committee. We've put a request in to get it reapproved.
Yeah.
It's just the taking of the blood, which is obviously you've got to be within 2 meters. So we've just had to bring in a new SOP for doing that. But Hannah, then we can get back in.
So Hannah, I think you're on the line. Do you want to talk to Helen about this? Because Hannah's having trouble trying to get Hannah Levis getting blood collected as well.
Can you hear me, Helen?
Hello, I can. Hi.
Hi. So yeah, you know Jess's project. She's supposed to be collecting blood from people in the Royal.
Yeah.
And we've got all the approvals from the Royal. Well, apart from they want the approval from the sponsor. But we're just waiting for them to sign it off as well. But they've been taking so long on the hospital side. So many people that I had to get to sign it off that I didn't know existed and things. But I'm hoping, when did you submit your sort of ethics?
So mine is just university ethics, and I emailed them a while ago, and then they sent me some forms to fill in, which I sent back, and they've got a meeting on Friday. But if you just email the whatever it is, research ethics generic email address.
Is that new ethics you're applying for, or just?
No, it's existing university ethics. So have you got University of Liverpool ethics as well as NRES?
Rec, yeah.
Yeah, so it's just the existing university ethics, and then I just emailed them to ask them what the situation was, and they've.
Have you got any news?
There's a meeting on Friday, Mick. They're going to make a decision.
Ooh.
I can send you afterwards, Hannah. Let me write it down because I'll forget otherwise. I can send you on what they've sent me.
Okay, yeah, that'd be great. Thank you.
I don't think they know what to do. I had to do a new cost assessment. So I spoke to Joe Middleton, and then I had to get an email from Pete saying that they were happy for us to take the blood on the premises. I guess yours is different, isn't it? She doesn't take blood in the building, does she?
No, it's the nurses at the Royal, and I suppose they would be dealing with them quite closely anyway. They like taking images of the eye and things like that for their routine clinical. So I don't think our SOP had to change that much because they would already be in close contact and following the social distancing things anyway.
Yeah, they brought in They've brought in a new cost assessment for off-site working, which you might need to fill in, but that doesn't affect us because it's all within the building.
Okay.
Can you two follow this up afterwards? I think.
Yeah, sorry, Rachel. You did start it. You did start it, Rachel.
Start it. You're quite right. I thought.
Thank
I'll email you after, Hannah. I've made a note.
Great, thank you.
Okay.
So Helen, are you ready to start?
I'm ready to
quite a few
start.
people now. So everyone, remember to put your questions in the chat, and we'll follow this up at the end. But Helen, take it away.
Thanks, Rachel. Apologies for the croaky voice that I've got today. Summer allergies are still bugging me. So what I'm going to talk about today is an overview of the work that we've been doing for the last 10 years, looking at applying different omics technologies to try and tell us more about neutrophil phenotype in inflammatory disease. And I've called this talk Inside the Chamber of Secrets for a couple of reasons. First of all, until fairly recently, neutrophils were a bit of a closed book. We didn't understand an awful lot about how they functioned. They're quite tricky cells to work with. You have to isolate them fresh from blood or synovial fluid every time you want to do an experiment. They're terminally differentiated, so you can't culture them in the lab and keep them going. And they're very tricky. If you handle them incorrectly, they will become inappropriately activated or die when you don't want them to. The other reason I've called it Inside the Chamber of Secrets is particularly when I go to clinical conferences, I tend to get a look like this when I tell people that I work with neutrophils. Poor old Ron Weasley here, a look of astonishment that anybody would want to work with neutrophils. I could, of course, use this guy as an example of that confused face that looks back at me. You'll know him if you're up to date with Twitter this week. He interviewed Donald Trump quite spectacularly and has become an internet sensation. Normally, this look of confusion as to why on earth you're working with neutrophils is followed by the classic line, oh, we just throw them away. For reasons that I've said, they're technically challenging, they die when you don't want them to. You can't transfect them, they don't go through cell division, so they're very technically difficult to work with. So hopefully, at least by the end of this talk, I'll have persuaded you not to throw your neutrophils away and that they are interesting little cells. So what I wanted to go through with you today, first of all, is just a bit of an introduction to neutrophils. I realise many of you won't have an immunology background. And then I'll go through the role in inflammatory disease and how we're trying to understand that further using transcriptomics, proteomics and metabolomics. So 3 short stories. We do have projects looking at the genome. I have a SEMA PhD student in Sheffield with Steve Renshaw looking at epigenetic regulation of neutrophils in zebrafish, and a clinical fellow, Rodri Smith, who works with me looking at variant analysis, but I'm not going to talk about that today. So what are neutrophils? Well, neutrophils are innate immune cells whose primary role really is to circulate the body, to look for invading microorganisms such as bacteria and fungi, and to phagocytose and kill them. And that's exactly what you can see happening here on the left of the screen. This grey cell is a neutrophil that's been incubated with Staph aureus bacteria in the lab. And then we've done an electron micrograph. And what you can see here is this dark black bacteria has been recognised by the neutrophil. It's engaged with cell receptors. Pseudopodia are forming around the bacteria to take it actually within the cytoplasm of the cell within a phagosome. And then these circular objects scattered through the cytoplasm are granules that contain proteolytic enzymes. These fuse with the phagolysosome and actually kill the bacteria. And that is their day job. They circulate the body looking to do this. As well as releasing proteases into the phagosome, they produce a massive oxidative burst and produce ROS, which is also cytotoxic. The NOX2 enzyme, which is an NADPH oxidase, rapidly reduces oxygen to perform superoxide and hydrogen peroxide. This is then metabolized by myeloperoxidase to produce secondary oxidants such as hypochlorous acid, which is a really effective microbiocidal molecule. Hypochlorous acid last week was in the press because in some parts of Asia, they've now set up these pods that you can walk through and be sprayed with hypochlorous acid to kill coronavirus. I won't be doing that anytime soon. So as part of this process, we get rapid generation of ROS and then rapid release of proteolytic enzymes within this phagolysosome that kills microorganisms. And it's these proteolytic enzymes that are implicated in tissue damage and disease. So when neutrophils are present, for instance, in joints in rheumatoid arthritis, we get release of molecules like neutrophil elastase, collagenase, myeloperoxidase, and these can damage the host tissue. They can cleave collagen. I've done some nice work with David Wilkinson recently that shows that neutrophil elastase will activate MMPs that can then go on to induce further damage within the joint. And these molecules are implicated in blood clotting. They're present in nets, which I'll talk about in a moment, and they're also autoantibody targets in certain autoimmune conditions. Neutrophils can also chuck out a huge array of cytokines and chemokines. These are molecules within the immune system that activate other immune cells. Many of these are pro-inflammatory, or they're chemokines that attract other immune cells to sites of inflammation. And whilst they may not produce cytokines at the same level as other cells such as macrophages, because they're present in such high numbers within inflamed tissues, they contribute significantly to the presence of these inflammatory molecules. So what's happening in inflammatory disease? So this is an example of rheumatoid arthritis. This is a joint. In rheumatoid arthritis, we get growth of an invasive inflammatory tissue over the surface of the joint. We get rapid accumulation of neutrophils within the joint. They become activated by immune complexes and by cytokines, and then they're actually start to release their ROS and proteases directly onto the surface of the joint to damage the cartilage. And as I've said, these they produce cytokines and chemokines, proteases that activate MMPs. They also activate proforms of cytokines. They can present antigen to T cells through MHC class 2, and they can actually shed receptors. So for instance, the interleukin 6 receptor is shed by neutrophils, and it can then activate neighbouring cells through trans signaling when those cells don't particularly express that receptor themselves. Neutrophils are a target of autoimmune inflammation. So in vasculitis, ANCA-associated vasculitis, the two main autoantibodies are PR3 ANCA, that recognises proteinase 3, a neutrophil protein, and MPO ANCA, myeloperoxidase. And autoantibodies against these two proteins expressed on the surface of the neutrophil will bind the neutrophil cross-link and activate the neutrophil to degranulate onto host tissue, causing damage to the delicate blood vessels. And neutrophils themselves may contribute to autoimmunity through the production of nets. Now, nets are important, so I'm going to talk about nets later on in the talk. Nets, if you've not heard of them, are a process where neutrophils, rather than phagocytosing and digesting bacteria, they will dismantle their granules, they will dismantle the nuclear envelope, and they'll start to dismantle the chromatin itself and assemble it into a structure that resembles a fishing net, which they can throw out of the cell to trap and capture bacteria at a site of infection. This DNA net is decorated with granule proteins and histones and many other proteins that are involved in bacterial killing. And it's been hypothesized for a while now that nets are produced in autoimmune situations. So for instance, in rheumatoid arthritis, we've got evidence of nets within serum and within synovial fluid. In lupus, we also have production of nets within inflamed tissues. And it's the proteins that are present on these nets, particularly post-translationally modified proteins, that generate autoantibodies. And we'll talk about nets later on. So I'm particularly interested in the role of neutrophils in rheumatoid arthritis. Rheumatoid arthritis, you can see here in these pictures, it's a chronic inflammatory disease, affects around 500,000 people in the UK. It's characterised by autoantibodies and elevated cytokines within serum and joint fluid, which activate immune immune cells and attract them to the surface of the joint. And autoantibodies are actually expressed on the surface of the joint, and this is what neutrophils will bind to and recognise to release the ROS and proteases within the joint. And if left uncontrolled, the joints will become swollen and deformed, as you can see here. And this is irreversible. This joint damage is irreversible. The bone and cartilage is completely destroyed, and the joints become deformed. And people can live with lifelong disability and pain with this condition if they aren't able to find a treatment that works for them. So we know from the work that we've done in the past that neutrophils are found at high abundance within rheumatoid joints. So if you've got a swollen knee and you put a needle in there and aspirate the fluid, it's got high numbers of neutrophils in there. And they have an activated phenotype, both in blood and synovial fluid. They've been activated in vivo to release their ROS and to release their granule enzymes. And what you can see here are some electron micrographs of healthy control neutrophil. You can see the nucleus, and these dark circular objects are the granules. And then in rheumatoid arthritis, you can see we've got a more ruffled plasma membrane, and we've got these large vacuoles and fewer granules within the cytoplasm. And what's happened here is this cell has become activated. It's formed these phagosomes within the cytoplasm, and the granules have actually degranulated into the phagosomes and also extracellularly. And we believe that production of NETs in rheumatoid arthritis and exposure of post-translationally modified proteins, particularly those with a citrullinacean modification, leads to the production of autoantibodies. And during my PhD, I showed that TNF production and NF-kappa B activation are elevated in rheumatoid patients, but that this returned to baseline in response to TNF inhibitor therapy. And this led to the hypothesis that blood neutrophils may actually be good reporters of underlying disease activity in rheumatoid arthritis. So the first story I'm going to tell you is the work that has been the main focus of me during 2 fellowships. First of all, an Arthritis Research UK Foundation Fellowship, and lately a Versus Arthritis Career Development Fellowship. This work was started with Hugh Thomas, who's now a postdoc in Manchester. And when we started doing this work, we didn't really know whether we could even do transcriptomics with neutrophils. As I say, they're technically challenging to work with. And actually, in relation to other immune cells, PBMCs, they have relatively low levels of RNA. So whereas you might be getting 1 million PBMCs, you're going to be wanting at least 5 to 10 million neutrophils to get the same yield of RNA. And we did a lot of work in the beginning to optimize our protocols. So you can't use an RNeasy Kiogen kit to isolate RNA from neutrophils. You need to use triazole chloroform precipitation. You need to use DNase digestion, etc., to make sure you're getting a nice clean signal. We did a lot of work as well to characterize what happens if you get the odd contaminating PBMC or eosinophil in your neutrophil preps, and concluded that really we need to be using ultra pure, 99% pure neutrophils for our RNA work. And the first experiments that we did were very simple. We just treated neutrophils with cytokines in the lab to see if we were getting a sensible signature, that we could get reliable RNA that made sense. And anybody that's done ingenuity analysis will recognize this activation of a TNF pathway. We'd actually activated neutrophils in the lab with TNF. So this was really nice that we got this nice clean signature when we did our informatics analysis. And this gave us confidence then that when we went to look at our rheumatoid patients, that we'd be able to interpret the results that we got with confidence. We did a lot of work as well looking at some key genes that are regulated by TNF and GM-CSF, and got a good correlation, a good pattern when we looked at using RNA-seq or real-time PCR. So then in the Foundation Fellowship, we moved on to look at gene expression in rheumatoid neutrophils. And I'm not going to share that story now, it's all published, but essentially we identified potential biomarkers of response to TNF inhibitors in these neutrophils. So we got 2 panels of genes which predicted either a good response to a drug or a non-response to a drug with high predictive value. And that has now gone into we've just getting the results together now from a larger clinical study of 50 patients. We also used RNA sequencing to identify a subpopulation of neutrophils called low-density granulocytes. These cells aren't expressed in healthy people to any high level. We found that they were elevated in rheumatoid arthritis. Actually, when you do density centrifugation, these neutrophils stay up with the PBMC layer. You can see them here in a mixed cell population within PBMCs. We obtained ultra-pure low-density granulocytes and use transcriptomics really to characterize them. They're expressing high levels of genes for proteases. They're still going through a cell cycle. They've got very delayed apoptosis and actually very low expression actually of inflammatory markers. So this is actually protein level data from a Western blot. They've got very low expression of TNF receptors, which is interesting for us as rheumatoid arthritis is considered a TNF-driven disease, that we had a subpopulation of neutrophils that were not responding to TNF because they didn't have receptors, suggesting there are other key cytokines and activating factors in RA that are activating these cells. So moving on now to my current fellowship, where I'm using some more advanced informatics analysis to try and understand what's regulating gene expression in RA neutrophils and to do some more validation experiments. We've got data sets from rheumatoid arthritis patients from 2 cohorts of patients, healthy controls and then healthy neutrophils treated in the lab with cytokines as our in vitro inflammatory model. And I've been applying different computational methods to really understand what's going on in these neutrophils, and I just want to give a flavour of some of the work that I've been doing. So the first thing that I wanted to do was just look on a global scale and do some multivariate analysis to see how different really rheumatoid and healthy control neutrophils are. This is looking at the whole transcriptome, which is about 14,000 genes, and you can see you get complete separation on this PLSDA model of rheumatoid and healthy control neutrophils, and actually we get an area under the curve of 1 based on 2 components to completely separate these transcriptomes. So this just tells us that they're different. It doesn't tell us what's different. So I next did some modular analysis. So this is based on a paper by Chaucer-Bell and colleagues, which produced modules of inflammatory genes. They compared gene expression in whole blood amongst several different autoimmune conditions. I think it was rheumatoid arthritis, lupus, diabetes, various others, and they generated these modules that you could group genes together to imply some functional regulation. And what we see actually when you run this through an R script is you see these are different modules and you can see the level of expression in healthy control and in rheumatoid neutrophils. This is the original cohort, and then we have a validation cohort. And what you can see is a commonality. So in rheumatoid arthritis, we're getting very strong modules relating to activation by type 1 interferon, antiviral signatures, RIG1, which is downregulation of interferon signaling. We're also getting activation of the inflammasome, albeit in a different position on this chart. And then we've got a lot of signaling pathways that are common. So we're getting an inflammatory response, we're getting modules relating to neutrophil activation, kinase activation, toll-like receptor signaling, and various pathways that are involved in regulation of transcription. So some similarities, but some important differences between rheumatoid and healthy control neutrophils. So the next thing that I wanted to do was to look at gene expression networks. So this algorithm identifies groups of genes that have similar levels of expression, with the assumption that genes that have similar levels of expression across your data set are co-regulated in some way. And we had a very high cutoff or low cutoff, 10 to the minus 20 p-value. To suggest that these are highly significantly regulated together. And what this has done is it's looked at the whole transcriptome of neutrophils in rheumatoid arthritis, and first of all it's predicted these 2 modules relating to regulation of gene transcription and regulation of protein modification and transport. We've got key transcription factors that we know actually are activated in neutrophils, the IF2, STAT5, NF-kappa B, RELA, which is part of the NF-kappa B family, and activation of signalling pathways related to GTPases, protein kinases, integrin, IL-8, JAK-STAT, TNF. So this is how we're seeing our neutrophils activated. Drilling down a bit further, we got 2 modules that were predicting activation by type 1 interferons, and PRR, which is pattern recognition receptors or toll-like receptors. Again, this is activation by factors such as LPS, lipopolysaccharides, interferons, activation of interferon response factor, transcription factors, and then finally 2 smaller modules relating to metabolism and leukocyte activation. So this is giving us a picture of what our neutrophils as a whole from rheumatoid patients look like and the main signalling modules that are activated. One of the important things that I wanted to do with this work is to look at the relationship between gene expression and clinical variables. So we've got very good clinical data on our rheumatoid arthritis patients. We know, for example, as well as things like age and gender, we know their disease activity score. So this is a 28-point disease activity score based on the number of tender and swollen joints. We've got measures of blood markers, so serolated protein, erythrocyte, sedimentation rate. We know the number of tender and swollen joints at the time of sample collection, and we know whether they've responded to therapy. So we're able to use these clinical markers to then build into a statistical model which uses random forests to identify those genes that are the best predictors of those clinical markers within the data set. And we've used a high cutoff of 0.95 to then look at the genes that are within those models to predict these clinical variables. And there's a lot of information here. This took an awful long time to run, did this, but essentially we're getting key messages. For example, the swollen joint score, so the number of swollen joints that you have at the time the sample is collected is regulated by things like STAT3 phosphorylation, apoptosis. We're getting a change in disease activity in response to therapy that relates to response to ROS and cellular stress. And we're getting a disease activity score post-therapy, which relates to things like, again, apoptosis and NF-kappa B signaling kinase activation. Now, some of this fits with work that we've done in the past. So as I mentioned earlier, we've shown that NF-kappa B activation relates to response to therapy in rheumatoid patients. So we know that many members of the STAT family are phosphorylated in rheumatoid arthritis and not in healthy controls. But really what the position that I'm in now is that this model and these predictions need validating in patient samples, which is on hold at the moment. But fortunately, we do have a lot of samples biobanked that I will be able to access as soon as we can get back in the lab to start working through validation of some of these predictions. Now, one piece of work that I have been able to finish off while I've been in lockdown actually is something that's been hanging around for quite a long time, which has been looking at gene expression in synovial fluid neutrophils. So back when I was a PhD student, before we had biologics like TNF inhibitors, we had a lot of patients who got chronically swollen joints and were regularly coming to clinic to have the fluid removed from the joints. So you can see, for instance, here, this is fluid from a joint. I didn't collect this. This image predates me, but these are 25 mil tubes, so we've probably got about 350 mils of joint fluid that's come from one knee here. Within this synovial fluid, you've got huge numbers of neutrophils, millions and millions and millions of neutrophils. And we know that these neutrophils have been activated. We know that synovial fluid is rich in cytokines and immune complexes that activates neutrophils. We know it's hypoxic, which is neutrophils like because it delays their lifespan. And we know from some of the work that that I did with Mandy Peffers a couple of years ago, that metabolites are different in rheumatoid arthritis synovial fluid compared to osteoarthritis. So for example, metabolites of glycolysis and TCA cycle are much lower in RA because we've got high levels of cellular respiration. And you can just see here, this is just a cytospin of whole synovial fluid. You can see there's a mixed leukocyte population, but the majority of these cells are neutrophils with their happy smiley faces, ready to damage those joints. So what I've been able to do during lockdown is to actually fully analyse some data set that I collected years ago. It's just been sitting, unfortunately, on the desk, one of those things to get around to. So we had transcriptomic samples from blood and synovial fluid neutrophils paired from the same patient at the same time. We've been able to look then at the transcriptomes of those neutrophils that are in the blood and then the changes that take place as they migrate into synovial fluid. So again, we've done this modular analysis. Lots of pathways are the same. Interestingly, what was different was that pathways relating to integrins and cell adhesion are massively downregulated when neutrophils migrate from blood to synovial fluid. This is this data overlaid onto an ingenuity pathway for leukocyte extravasation. What you can see is huge numbers of green genes. These are being downregulated in synovial fluid. And this is suggesting that neutrophils, once they migrate to the joint, actually stay there to do their damage. And the pathways that we see upregulated, we've got antigen presentation, so presentation of MHC class 2 to T cells, production of chemokines, HIF1 alpha signaling, that's hypoxia response, transcription factor, and downregulation of these adhesion pathways. And here, this is just the expression of some key chemokines that are involved in activating other arms of the immune system. You can see these are massively expressed in synovial fluid. And indeed, we've shown in the lab that synovial fluid induces almost 100% neutrophil migration in the lab through partially permeable membranes, and delays neutrophil apoptosis and extends the lifespan. We were also able to show that synovial fluid can stimulate production of nets. So these are nets here. This is untreated neutrophils, and this is neutrophils that have been treated with a positive control PMA. You can see huge production of this DNA net, and this red staining relates to myeloperoxidase, which is decorating the net. When we stimulate with synovial fluid, again, we get huge production of myeloperoxidase positive nets, but we're also getting high levels of citrulinated histone 3, and that's a protein that's undergone that chemical modification to express citrulinated peptides. And these are highly recognised by the immune system as foreign peptides. And again, Ingenuity has predicted some key pathways that maybe regulating production of nets in synovial fluid, and this is something that I've been really trying to take forward in grant applications, unsuccessfully, unfortunately. So this summarises the first piece of work, the transcriptomics. What we were able to show was that blood neutrophils circulating in blood vessels have an interferon-induced gene expression signature. They undergo delayed apoptosis. They've got increased signalling through pathways such as JAK-STAT and NF-kappa B, which are activated by cytokines. They show levels of oxidative stress, and they have an altered metabolism. And what happens when they migrate out of the blood vessels into the joints is that we now have a change in phenotype. We have expression of MHC class 2, we have delayed apoptosis, they become activated by immunoglobulins, which they will phagocytose or they will bind to on the surface of the joint. This induces degranulation and production of ROS, and also production of nets. And we have massive expression of chemokines, and these chemokines that are being expressed by neutrophils are able to activate all arms of the innate and the adaptive immune system. So they're really central to the process of driving inflammation within the joint. So I'm going to move on to another story now. This is a story of proteomics. This work was carried out by Eleanor Chapman, who many of you will remember. She was a Wellcome Trust Seed Award postdoc that worked with me for 18 months. And Max Lyon was a clinical MRes student that worked with myself and Eleanor on this project. And what we wanted to do in this proteomic study was to really try and understand nets further. And to do that, we wanted to look in two different diseases. So rheumatoid arthritis, which I've introduced to you already, chronic disease of the joints, but also lupus. Lupus is much rarer than rheumatoid arthritis, but it's really a terrible disease and can be life-threatening in many conditions. It's characterized by this rash on the face. Patients get damage to the skin, they get damage to their kidneys, they get vasculitis, there may be brain and cardiac involvement. Patients can be really poorly who've got rheumatoid arthritis. And in both, lupus, in both these conditions, we get chronic activation of neutrophils and production of ROS, but there are some differences. So in rheumatoid arthritis, apoptosis is delayed, whereas it's increased in lupus. And we get production of nets, and we believe that exposure of these citrullinated proteins in rheumatoid arthritis causes production of autoantibodies directed against citrullinated proteins, so anti-CCP or ACPA, which is a clinical diagnostic for rheumatoid arthritis. And again, in lupus, we get production of nets and exposure of these intracellular proteins. And in lupus, we get autoantibodies against double-stranded DNA and anti-nuclear antibodies, such as anti-histone antibodies. So a highly hypothesized role for nets in the development of autoimmunity in these two conditions. So as I mentioned before, nets are these extracellular structures of DNA and protein that are released by neutrophils. And we believe this to be a normal process during infection. And there are some animal models from Paul Cubes' lab in the States, where they've used an infection model and they see rapid production of nets within the liver, for instance, and these are then able to trap bacteria so that other neutrophils are able to come along and kill them. So this is what nets look like. This is a zoomed in image. These are nets that we've generated in the lab from 3 different individuals. We've got staining of the nucleus in blue, so this is DAPI staining of the nucleus, and then antibody staining against myeloperoxidase here in red. And you've got cells that haven't undergone net production there, and you've got cells that have undergone net production there. And you can even see on this middle image a cell body that has lost its DNA because this has been extruded by the cell. And now there are at least 2 ways that neutrophils can produce nets. We call these suicidal and vital. So suicidal netosis involves activation of the NOX2 enzyme, that enzyme I mentioned at the beginning that's involved in production of ROS. And during suicidal netosis, the nucleus is completely dismantled, the chromatin is loaded with proteins, the cell bursts and the nets are thrown out of the cell. In vital netosis, NOX2 is not involved. We believe it's mitochondrial ROS that trigger vital netosis, and there is good evidence to suggest that the nets are released, but the cell remains viable and will actually crawl away and carry on without its nucleus phagocytosing bacteria. It's pretty cool when you see that on a movie. So what we wanted to do in this study was really compare these 2 modes of net production. As I said, NOX2 dependent nets involve production of ROS, release of elastase to degrade the histones to release the net. In NOX2 independent, we've got activation of mitochondrial ROS and activation of PAD4, which is a really key enzyme when we're talking about nets, and particularly in rheumatoid arthritis, because PAD4 is the enzyme that's involved in chemical modification of arginine to citrulline. And it's citrulline and citrullinated proteins that are recognised in rheumatoid arthritis by ACPA or anti-CCP autoantibodies. And a nice study that came out of the Netherlands, I think, a couple of years ago, was looking at net debris in serum from different autoimmune conditions, and they concluded that the majority of net debris in lupus and rheumatoid arthritis came from NOX-independent nets. So they weren't nets that were produced in response to ROS, they were nets that were produced in response to PAD4, in comparison with psoriatic arthritis, which is more ROS-dependent net production. So we used two agonists in this study. We used PMA, which induces NOX2 dependent nets, and we used A23187, which is a calcium ionophore that induces NOX2 independent nets. And we found, yes, that they both activate net production in lupus and rheumatoid arthritis. So then we did a lot of optimization of our protocols, and Eleanor was able to gently digest away the DNA containing proteins away from the cell bodies to then take that into a clean prep where she was able to remove the DNA and do proteomics on the proteins that had been lifted from the nets. And what we found actually in both RA and lupus, there was a core of about 300 proteins that was the same, independently of how we activated net production. But actually, again, there was about 180 proteins that were different depending on whether we'd activated the nets with PMA or with A23187. And this was similar actually between RA and lupus. And if we compare PMA-induced nets here in RA and lupus, actually there's only 8 proteins that are different from almost 500 proteins. The same again with A23187. So we concluded in this study that the process of net production is actually, it's different whether it's NOX2 independent or dependent, but it's virtually the same depending on the background of where the neutrophils actually come from. There were some differences. So there's some examples here. We did some immunocytochemistry.
Hello?
You can see, hello?
Yeah, speaking. Yeah.
Oh, can you turn your microphone off, please, Nordine?
Okay, that's fine. Yeah.
Sorry. So here you can see staining of MMP8
Yeah,
is higher in our rheumatoid arthritis nets. And then we've got PAD4, this key enzyme that's involved in citrullinación of proteins, is highly expressed in both rheumatoid arthritis and in lupus nets. Histone 2B, which is a known autoantibody target in lupus, is highly expressed in our lupus nets. We were also able to get a more understanding of post-translationally modified peptides, particularly in lupus. We saw high post-translationally modified histones. So histone 3 was really interesting. We were getting acetylation, methylation, and citrullinación of histones, which is interesting because a lot of post-translationally modified histones are autoantibody targets in lupus. So to kind of summarize the work that we did here, in rheumatoid arthritis, we saw levels of MMP8, MMP9, elastase in our nets. These are implicated in activation of other MMPs, degradation of collagen. We got citrullinated histones, again, recognized by ACPA antibodies. And citrullinated histones, as well as vimentin and alpha enolase, are recognized by this particular HLA subtype, HLA-DRB1-040104. This is known as the shared epitope. It's a HLA subtype that genetically predisposes people to developing rheumatoid arthritis. And if you have this genetic subtype and then make certain lifestyle choices, such as smoking, which massively induces citrullinación of peptides, you're about 50 to 60 times more likely to develop rheumatoid arthritis than somebody who doesn't have this shared epitope. In lupus, as I mentioned, we saw huge numbers of post-translationally modified histones, particularly acetylated, methylated, citrullinated histones. We also saw LL337, which is a well-known autoantibody target in lupus. It's well characterized. It's been shown to be elevated in lupus serum and skin. So to summarize the proteomics, in this work we showed that NOX-dependent and NOX-independent nets contained different proteins, but that this pattern was similar irrespective of whether the background of the neutrophils was from disease or healthy. We did find some proteins that were different between RA and lupus nets. Some of these were interesting and relevant. For instance, we saw granule enzymes higher in rheumatoid arthritis and histones higher in our lupus nets. We got a lot of information on post-translationally modified proteins, particularly histones, and we think that these differences between lupus and RA nets may lead to better understanding of the role of nets in disease, and I really think nets could be an interesting therapeutic target to take forward. So the final piece of work I want to talk about is metabolomics. This is work that was started by Susama Choksawata-Nazagul. Susama was a PhD student on the joint programme with Chulalongkorn University. She stayed on after her PhD to do a postdoc with me. This is now being carried on by Mick Fresneda. Many of you will know him. He's my current PhD student, just coming to the end of his first year. And we've been working on protocols to look at metabolism in neutrophils. Now, metabolism is really hot right now in immunology. It's basically biochemistry rebadged, but by calling it immunometabolism, suddenly as a biochemist, you're the person at conference that people want to talk to. Many of you may have seen Luke O'Neill on Twitter or even on some of the TV programs talking about immune function in coronavirus. Luke is a superb speaker, and he is an absolute metabolism specialist, and he's everywhere at the moment because metabolism is super hot. Because it's been determined that metabolism can really rapidly switch on or off immune cell function in inflammation. So T cells and macrophages particularly, their function is regulated when they migrate from the blood into inflamed tissues through switches in metabolism. And this is what metabolism looks like for anybody that didn't do biochemistry, and for everybody that did do biochemistry, that's what metabolism looks like. It's really, really complicated, intricate pathways talk to each other all the way through this. And we know from work that's been done that metabolites, for instance, in synovial fluid can enter a metabolic pathway halfway along and, for instance, activate an inflammasome or activate cytokine signaling. And we know from work that was done 40 years ago when it was biochemistry that metabolites of glycolysis, for instance, can be redirected into the pentose phosphate pathway to fuel ROS and NET production. So we hypothesized that metabolism could be a druggable target in rheumatoid arthritis. So when you do NMR metabolomics, this is kind of what you get out of it. Apologies to Mick and Marie if you're listening to this. I hope I do this figure justice. This is an NMR spectrum from a neutrophil cell extract. You've got several cell extracts overlaid here, and each of these peaks relate to not an individual metabolite but an individual chemical structure on a metabolite, and each metabolite may have several different peaks relating to the chemical structure of that metabolite. And this gives a metabolic fingerprint of the sample. And the challenge is to extract data on the metabolites that you have in your solution or your cell extract from these spectra. So just zooming in, you can see these metabolic spectra can be broken down into tiny different peaks relating to different chemical structures. And essentially, if you have a student working in the NMR centre creating their pattern file, they're cutting this up into tiny little chunks and then identifying using a library what each of these peaks relates to. And if they're doing that, you probably won't see them for a couple of months because it's incredibly laborious work to get this done to Marie's high standards. So when we started doing this work, again, we had to do a lot of optimization of our protocols. Neutrophils are not cells that you can use with a kit mentality or take somebody else's protocol. So we found, for instance, that just something as simple as having HEPs in our buffers was a major problem. Even though we'd done wash steps, we got contamination of HEPs, and it was clouding our signature that we were getting from our actual neutrophil pellets. And we found through the work that we did that actually just by including something as simple as a heat shock step when we got our neutrophils to a pellet, increased the yield of metabolites. So you can see here, and the top line doesn't have a heat shock step, and the bottom line does. You can see we're retaining metabolites here that are being lost when we don't have that heat shock step. So really important work that Susama did during her PhD there to get these protocols established. And the first paper that we had out of this was really a protocol paper, but we showed that neutrophil metabolism could be rapidly altered by hitting them with high amounts of PMA that just blasts the neutrophils and switches them right on. Within 5 minutes, you're generating a very, very different metabolite profile. You're getting rapid production of NADPH, lactate, etc., rapid activation of NAD metabolism and glycolysis just by switching these cells on. They very, very quickly respond to activation. So then we wanted to look at what was happening in rheumatoid arthritis. And this is just some preliminary data from the grant application that brought in mixed scholarship, where we showed that rheumatoid neutrophils have got a different metabolic profile to healthy control neutrophils. And you can see we've got some metabolites that are higher in RA and some that are higher in healthy controls. And this is where Mickey's at really now with his PhD. He's got lots of samples collected before we went into lockdown, and he's just itching to get back into the NMR centre and start to analyse his own samples to really drill down into these rheumatoid and lupus actually samples to see what's different from healthy controls. So Mick's been working on a data set that Susanna generated before she finished, looking at the effect of JAK inhibitors on neutrophil metabolism. JAK-STATs are downstream regulators from cytokine receptors. They induce rapid activation from cytokines such as IL-6, GM-CSF, interferons, and they're a new target of biologics in rheumatoid arthritis. So baricitinib, which is a JAK1/2 inhibitor, tofacitinib, which is a JAK3 inhibitor, have been shown to be really effective in treating rheumatoid arthritis patients to getting inflammation under control. So we wanted to understand what was happening to neutrophils in response to these JAK inhibitors. So we've got neutrophil extracts from rheumatoid cells that have been treated with either baricitinib or tofacitinib. And what Mick's been able to show is that they are different from untreated neutrophils, but actually baricitinib and tofacitinib appear to have quite similar modes of action. So if you look at them on their own, they're different from untreated cells, but they're quite hard to distinguish when analysed as together against untreated cells. But what he's been able to pull out are just over 70 metabolites that separate either of those drugs from untreated cells, and then the common metabolites that appear to be regulated by both of these are listed here. So we've got things like glycine, aspartate, homocysteine, glutamate. And then if you look at what's different between baricitinib and tofacitinib, again, we've got by baricitinib formate, histamine, histidine, glutamate, whereas tofacitinib appears to be levels of NAD, xanthine, glucose. So we need to do further analysis. We need to do some pathway analysis. We're not there yet. This is literally this week's data, but I can see that here these metabolites are feeding into ROS production, whereas we don't see them over here. So there could be something very interesting there, and that's what Mick's been doing during lockdown. So to summarise the metabolomics, we think it's a very powerful tool to identify differences between biological samples. You can analyse lots of different biofluids, so it could be a cell extract, could be serum, could be synovial fluid, could be urine. It's non-destructive and untargeted. And what's interesting about NMR is that you can identify a metabolic fingerprint. So even if you don't know what the metabolites are, if that metabolite is present in your patient and absent in your healthy control, it can still be a diagnostic marker because of this metabolic fingerprint on that spectra that's produced. And this is something that we're pushing forward with now in the lab. So to summarise, last slide, I've shown that omics analysis of neutrophils is possible, generate some really interesting data, but you have to optimise your protocols. Neutrophils cannot be treated as any other cell that you would culture in the lab. They are tricky to work with. You have to optimise everything properly. They do have an altered transcriptome, proteome and metabolome in rheumatoid arthritis and lupus actually, compared to healthy controls. They're interesting cells. We need to learn more about them. And the hope is that modelling these changes will provide novel insight into the phenotype of neutrophils in inflammatory disease. So last slide, people to thank: Mick and Marie, my current team. Marie's the manager of the NMR Centre. We work very closely together. I've name checked everybody in the neutrophil group. We've got clinical collaborators at Aintree Hospital that have been key in helping collect and process some of these samples. Collaborators in the proteomics, the computational biology facility, the clinical trials centre, and the imaging suite, and the people that funded the research. Thank you.
Oh, Helen, thank you very much. Oh, that was very quick, closing down your slides.
Put it down, I'm ready. You can see me instead of my little tiny third bedroom.
You've got loads of questions in the
excellent.
chat box. So George starts off by saying, why does RA manifest only in joints?
It doesn't.
You've got neutrophils everywhere.
It doesn't manifest only in joints. They have, it's mainly in joints, and that's because joints, for example, express high levels of the citrulinated vimentin and alpha enolase that patients get chronic inflammation, so they've got an increased risk of stroke and cardiovascular disease, for example.
George, you've got a couple of other questions. Do you want to come online?
Yeah, I've forgotten what I have asked.
Okay, the other one is, can we stop neutrophils entering the synovial fluid?
Not at the moment, we can't. Possibly if you had some sort of inhibitor against neutrophil migration, so an integrin inhibitor or something like that. The problem you've got then is that if you get an infection, are you going to inhibit them from migrating into sites of infection? So it's all a balancing act with neutrophils. You want them to still be able to do their day job if you get a chest infection, but you don't want them to go into the joints.
So neutrophil mice that don't have neutrophil-less mice, they survive pretty well. So why can't we do a transient knockdown of your neutrophil for a while you have a flare, for instance?
Possibly. That is definitely something that potentially could be done. What's interesting I didn't touch on neutrophils in mice. Neutrophils in mice are quite different. So they express different cytokines and different receptors. They're not a perfect model of human neutrophil function. What is exciting about inflammatory arthritis models in mice is that if you induce inflammatory arthritis, the first cells that get to the joints are neutrophils, so they seem to be initiating disease. But also there is a serum transfer model of inflammatory arthritis. So if you've got a mouse with rheumatoid arthritis, you take the serum, you inject it into another mouse, the other mouse develops arthritis. If you inject that serum into a neutrophil deficient mouse, they don't get arthritis. So yes, you're correct. There's good evidence that if we could knock out neutrophils, it may be a temporary solution to halting.
But I thought that the neutrophils didn't last very long, and they just get more and more getting stimulated. So if you knock them down, wouldn't you just get more?
Possibly, but it could be a short hit, might be all you need to break that chain, possibly.
Mandy, you got a question about RA and OA.
Yeah, so I found the same as George. It's like, what did I write?
Hi, Rumi.
Oh, sorry. Should I read it? Synovial fluid stimulates production of nets. What was the source of synovial fluid? Is it dependent on whether it's RA or OA?
It was RA synovial fluid, and I've not done the experiments with OA synovial fluid because it's really difficult to get hold of.
What about normal synovial fluid?
Oh, you can't get that.
I can get that. We'll just try some horse stuff.
Horse stuff? We could try it with horse stuff.
Synovial fluid, see if it has the same effect.
We could try it with horse stuff, definitely.
Yeah. Is it something in synovial fluid, or is it something in RA synovial fluid? That's my question.
It's probably immune complexes in RA synovial fluid. Probably.
So we've done lots of experiments where we've treated chondrocytes with synovial fluid. And then looked at the effects. So it'd be quite interesting to see what the effect of normal synovial fluid is as opposed to the RA synovial fluid.
Yeah, definitely. So you can't really get normal synovial fluid from humans. But yeah, if you can get it from horses, great. I don't know what human neutrophils will think of it, but we can try it.
Yeah, I've also got some normal synovial fluid bank that we bought a few years ago.
It's full of ethanol, isn't it?
Yeah. you think that'll have an effect? The ethanol?
Might do.
Treat with ethanol as well and see what the effect is.
Could be all drunk.
Denai, do you want to ask your question about RNA extraction kits?
Yes. Do you have any idea why the extraction kits were not working? Is it related to the type of tissue, type of cell, or is it related to the amount?
I think it's the amount and the fact that you need so many neutrophils that you're just getting your columns clogged with everything else that's in there. So what we do actually is we use triazole, chloroform, and then we precipitate the RNA from the chloroform, and then we dilute that in water and then pass that through a column and do an RNA cleanup there that way.
Okay. Okay.
But it's quite useful because you can keep the stuff that's left behind in the triazole and do protein extract.
Yeah. I was just wondering because we've had issues with isolating RNA from laser captured samples.
Mm-hmm.
So if it was just an amount issue, maybe then a normal extraction rather than very specific columns might be better.
Mm-hmm. Yeah, I think it's just they've just got lower levels of RNA, so you need more of them, and it's just clogging the columns.
Yeah. Okay. Thanks.
Thank you.
Sunil, do you want to ask your question about the beautiful electron micrographs?
Ooh.
Oh yes, thanks. Hi Helen, thanks for
Hi.
good talk. Earlier on in your slides, you showed a nice picture of neutrophil. I presume that was picked up on an electron micrograph.
Yes.
Now, it's got me thinking because I've been working on electron micrographic images of heart tissue, cardiac tissue, and this was not the place I was expecting to see immune cells, but I've seen structures similar to that, and I assumed they were macrophages. I was wondering if there was any way to differentiate between a macrophage and a neutrophil and also a monocyte on an electron micrograph.
The size would definitely differentiate between a neutrophil and a macrophage, possibly the nucleus as well. It really depends how your tissue's been cut, whether you get a clear image of the nucleus.
Well, the images we've got is a stack, so this is 3D electron microscopy. So we can look at 3D structures, but they seem to share these vacuoles that you mentioned about. Apart from the vacuoles, the monocytes tend to have these dark patches. Well, vacuoles are quite dark on electron micrographs. So we just look for the nucleus then.
Yeah, so I've not intensively studied electron micrographs of different immune cells, so I'm probably not the best person to advise on that. But I could take a look at a picture if you got something you wanted to send me. But I don't know that you would definitely know whether you'd have to do some sort of labeling. Nanoparticle, you can label, can't you, like nanoparticles that come up on the EM? I'm not sure.
Yes, but what we've done is juiced up the whole tissue and looked at proteomics, and we've got macrophage marker CD163. If there was any marker for neutrophils, I would be happy to look through my list.
Yeah, so CD15, CD16 is on macrophages as well, CD10, so probably CD15, CD10.
Super, thank you.
But if you want to email me,
yes,
email me please.
I'll do that. Thank you.
Okay.
And Vanya, do you want to ask your question?
Yeah, thanks for a nice talk.
Hi, Vanya.
Hi. I was just wondering whether when you do RNA-seq and proteomic experiments, whether you isolate the neutrophils directly from patient samples, or are you able to actually culture them? And if you can culture them, how long for?
So we tend to work with freshly isolated cells because we want to capture that, capture them as fresh as possible from when they came out of the person to capture that image. You can culture them. So we've taken healthy neutrophils and you can culture them for up to 24 hours really and do RNA sequencing, but the gene expression will change over time. So it depends what your question is. Are you interested in gene expression in response to an activating factor? So you might want to do a few time points. Or are you interested in gene expression in a patient compared to a healthy control, you'd want to do straight away 0 hours, isolate your cells, zap them into triazole straight away.
And do you still see that if you were to keep them for say 24 or 48 hours in culture, would you still see the differences in the ROS and cytokines between the control and the RA patient cells?
I haven't done those experiments, but I doubt it. So after 24 hours, if you don't treat them with a cytokine, at least 50% of them will be apoptotic. So they will have dismantled everything and got rid of all their RNA.
Is that just the nature of neutrophils?
Oh, yes.
Okay.
That's why people look at you with that face that I showed at the beginning, that look of horror when you say you work with neutrophils.
Because I was just thinking, is there any way, for example, that you could sync the neutrophils and then see whether after PMA or other treatments, they show more or less ROS or cytokines as in a time of day manner? Just because I know that a lot of people in Manchester are working with inflammation and these kinds of things, and the inflammation is like a big...
Yeah, so there are people looking at circadian rhythms. So the person you want to look for is Andres Hidalgo. I think he's in Madrid, and he's done a lot looking at circadian rhythms. And I think what they actually do is they take blood at different times of day rather than synchronizing them in the lab.
Okay.
They'll look at different times of day. So definitely net production is circadian, cytokine production is circadian, some gene expression is circadian.
Oh, really? Okay.
Yeah. So we've checked in our data because we tend to, when these stories started coming out, I panicked thinking, oh my God, did I get half these patients from the morning clinic and half from the afternoon clinic? But there isn't anything in our data, but receptor expression will change, definitely. So there was a really good review last year, but if you look for Hidalgo, Andres Hidalgo, he's the guy to look for.
So is this something that you can control in your experiments? Like, I mean, do you then decide that you will take patient samples at a particular sort of like time frame in the morning or afternoon? Or?
We try to target the same clinics.
Okay.
So for instance, the early arthritis clinic of new patients is always on a Wednesday morning. So you're going to get samples between 9 o'clock and 11 o'clock. And within that window, you're probably safe. And we do the same with our healthy controls. We generally take them in the morning because we want to do an experiment that day.
Okay, thanks.
Great. Any other questions, anybody? Well, Helen, extremely interesting and very different from anything I really understand.
Thank you.
So it's really nice to be able to understand this a little bit better.
Rachel, I examined a PhD yesterday looking at neutrophils and inflammatory eye disease, so maybe it's not that different.
Well, I never know. It might well be something interesting.
I learned definitely not to Google terms related to inflammatory eye disease.
Get horrible pictures.
Oh, yeah. I'll stick with joints.
Thank you very much. And,
thank you.
Thank you, everybody.
Thanks for all your questions. Bye, everyone.